Good morning. Hopefully you guys got the last video over the female side on reproduction. Today I got some slides and some videos on the male side of our reproductive tract. Hormone and endocrinology systems work very similar, and I'll explain the differences as we go, but thought we'd go over some of the basic anatomy of the bull. Obviously we got the scrotum here and the testes lies within it. You can see here the epididymis, the head, the body, and the tail of it. That's where sperm maturation happens, and then as it leads up, the duct deferens or the vas deferens to our secondary sex glands, which are really important in terms of the extension of our ejaculate. That's the vesicular gland, the bulbourethra, and the prostate. They're going to add to that through ejaculation and keep those semen cells happy and healthy as they make their way to the ampullus misjunction, which we talked about yesterday on the female side. So they have to work their way through the cervix and down that uterine horn to the place of fertilization. For our test, we need to know those parts and some of the functions of each one of them. So hopefully you guys have that on your video. Obviously, the scrotum is super important to keep the testes cooler than body temperature. We got to be about 4 to 6 degrees cooler, so those semen cells are happy. If we get them up to body temperature, they only live for a short while before they die or they become non-functional or non-fertile. In some cases, we run into cryptorchids or cryptorchidism, which will affect that semen quality. And when you have that, your semen is typically dead. Your bulls or your stallions will act the same because the temperature doesn't affect the steroid hormones like testosterone. So testosterone will still be produced, your animal will still act like a male, but will not be fertile. So therefore, it does you no good to turn them out to breed cattle or horses or pigs or whatever if it's a cryptorchid. Now, sometimes we have bilateral cryptorchidism, which means one of them will extend into the scrotum while the other one stays up in the body cavity. In this case, your animal still has a chance to be fertile. Obviously, everything that's ejaculated from the testes that's up in the body will be dead and deformed, where you still have functional sperm cells in the testes that is in the scrotum. Not a good situation for a breeding animal just because it drops your sperm production by half. So if I'm trying to get maximum conception rate, just not a good situation. If I only want to get 1 or 2 animals bred out of them, then it would probably be successful. As we look at this, you can see how the spermatic cord drops into the testes. You can see those arteries. It's called the piniform plexus, and this helps dissipate the heat. Obviously, you can see as our arteries and veins come down to feed the testes, there's very few that are around the testicle. We need a blood-testis barrier. Most of you know why, because when the male is born, the testes are there, but there are no sperm cells. We have cells called the Sertoli cell that are in the testes, and as at puberty, it kickstarts these cells to start producing sperm cells. So the immune system does not recognize sperm cells as part of the body. So if we have blood around our sperm, it automatically tries to kill it. That's why we have limited number of arteries and veins that are feeding into the testes. This is some of that control. You can see how the brain is still working with the rest of the male reproductive tract. We still have the hypothalamus releases GnRH. It still affects the pituitary releases FSH and LH. FSH stimulates sperm production, so it kickstarts the Sertoli cells. LH kickstarts the Leydig cells, which are producing testosterone. Testosterone is its own feedback mechanism. As testosterone gets too high, the brain cuts it off, boom, it drops. So it goes up and down constantly through the year. For sperm production, as we get more sperm in there, we don't have sperm in the bloodstream, so the brain doesn't directly know how many sperm cells there are. But as the Sertoli cell produces sperm cells, it also produces this hormone called inhibin. And inhibin is the feedback mechanism to the brain to know when the brain needs to produce more sperm cells and when it needs to cut off production. Also controls those muscles that raise and lower the testes within the scrotum. So this tunica dartis muscle, which helps that contraction and release, is also controlled that way. We have these sperm cells here now. So in the head, the body, and the tail, you can see the sperm come into the epididymis fully formed. They're half the genetic material at this point. They are a gamete, but they have not went through their complete differentiation. We don't have functional tails yet until we get to the tail of the epididymis, where we then have fully functional sperm cells. If we just took sperm cells right out of the epididymis, we would run into some issues in terms of fertility and them being able to fertilize, because they have not went through capacitation, which happens at the time of ejaculation. Sperm cells have to go through capacitation so that they arm the enzymes that's on their sperm head, so that they are able to penetrate through the layers of the egg and cause a fertilized pregnancy. This is just an actual picture of a testicle. You can see here, this is the testy. The testy is primarily made up of seminiferous tubules. Those seminiferous tubules are the majority of that testy. You can see the epididymis that lies here on the outside of the testy that's connected to it, and obviously this feeds into the head of that. So it goes from the seminiferous tubules to the ret tubules into the epididymis, and then feeds down to the tail of the epididymis, where semen cells are stored before ejaculation. Just a little better picture of it cut open. You can see those seminiferous tubules in there. If anybody's had Rocky Mountain oysters or calf fries, that's the main part of them. So we talked about fertilization taking place in the oviduct. There are fertilized embryos that we glanced at already. We hope that cow has recognition of pregnancy, because if she does, then she will keep her CL, and that CL will help maintain that pregnancy throughout its full term. You can see here we have some embryos that have been graded, and of all of these, they are all fertilized and ready to implant. These, you can see they are hatched, or they have a crack in the zona pellucida, which means that this cell mass is ready to float out and then attach to the uterus. This is a pregnancy. You can see the layers of it, the allantoic cavity, the amnion, the chorion. We have amniotic fluid in here around the baby. You can see the connection point here, which would be the umbilical cord that maintains that pregnancy throughout the time. In terms of some of our new technologies, here we have artificial insemination, where we are actually AIing a cow. You go through rectally with one hand, you pass a pipette through the cervix, and then you drop the semen in the uterus, and it swims to the oviduct to cause a fertilized pregnancy. In embryo transfer, we superovulate the cow, so she ovulates a lot of eggs. We then breed all those eggs, and 7 days after that, we go in with a barred Foley catheter, and we flush fluid into the uterus. We swirl it around, we flush it out into a dish, where we collect those fertilized embryos that are 7 days old, and we can then transfer them into recipients. So one cow could have 10 or 20 calves out of each cycle versus one calf. With this is the first set to do like in vitro fertilization. This is oocyte retrieval. Oocyte retrieval starts the same way as embryo transfer, but we do not fertilize those eggs. We superovulate her, and then we go in before she ovulates, and we take a needle and we pluck them into each follicle, which you can see here, and there's a vacuum pump that sucks the egg out. They are not fertilized at this point. Then you need to go under the hood, and you can see here how we do in vitro. So we go under the hood. Right here is a vacuum that holds that egg stable. Here is a needle that's going to penetrate the zona pellucida and inject the semen cell into the egg, and hopefully cause fertilization, and then we'll grow it for 5 to 7 days in a sterile, hooded environment. Cloning is a bit different. We have to take a DNA strand from an animal, we grow it into an embryo, and then we put it into a host. The host then has the clone of whatever we took that DNA strand from. In this case, this is Dolly. This was the first cloned sheep. Obviously, the clones are genetically identical, but they have not had the same environment as their cloned parent, so therefore the animals will not act and be identical. There are some disadvantages of cloning. In some cases, if we clone too much, we then have a genetic population with no diversity, which means that a disease could come along and wipe out the whole population. Hence COVID-19, right? If we didn't have some diversity in our population, we would all be subject to sick and ventilators as some of the population that's had that happen to them. So not everybody is going to react and respond to each disease in the same way. Therefore, genetic diversity is a good thing. Cloning is a good thing in certain situations, but should not be our only source of reproductive methods. Anyway, I hope this gave you a little more light. I hope you watched this video, and I will be posting a little bit more information on Thursday, and on Canvas you will find the final that's due next week.